Are we? Wonderful. Okay, so we do have a good turnout today. Welcome, everyone. I think everyone here is familiar with Dr. Kelly Jordan-Sciutto. She's a professor of pathology in the Department of Oral Medicine in the School of Dental Medicine, where she wears many hats. She also serves as Associate Dean of Graduate Education, Director of Biomedical Graduate Studies, and co-director of the Penn Mental Health AIDS Research Center and the Penn C4 Scholars Program at the Perlman School of Medicine. Beyond that, she's also a fabulous mentor who's committed to enhancing the educational and mentored experience of graduate students and increasing diversity and equity through an inclusive culture. And I personally won't forget that she was also one of the first people to ask me a question at my very first oral presentation at ISNV. So her research focuses on the molecular and cellular mechanisms of neuroinflammatory induced CNS damage during HIV infection. And today she'll be presenting on genetic variations that may contribute to HIV-associated CNS changes through altered stress tolerance in neurons and glia.
Thank you very much for that introduction and for the fond memories of that specific ISNV meeting, which was awesome. So thank you very much. I'm really looking forward to feedback today on this project because it does incorporate some genetics, some of the cell molecular biology, as well as some sort of organellar stress, which is all things that I'm interested in. But with this intersection, I think there's lots of room for advice from the community so that we make sure that we're doing the best things that we can. So I'm just putting this slide up because I feel it's like I'm obliged to start here, even though I know that everybody already knows this. Are you guys seeing the panel of everybody on my screen or no? Just making sure.
No, just a slide with a pie chart, with two pie charts.
Perfect. So, so in this, just as a reminder, HIV-associated neurocognitive disorders still persist, particularly in the ongoing cohorts that are at Charter, as well as Max, Wise, and as others. So while I think there is a growing trend in terms of intervention early that may change the way these pie charts look, we do know that there are, at least among those who have been living with HIV prior to that sort of change in the clinical paradigm, a number of, about 50% of people were continuing to have HIV-associated dementia, minor cognitive disorder, and or asymptomatic neurocognitive disorder. And so the idea here is that we've done a really good job in terms of reducing the people with more severe or functional deficits with HIV-associated neurocognitive disorder. But we still have a number of individuals who fall into this asymptomatic neurocognitive impairment that can be measured with tests, doesn't impact daily life, but maybe at a higher risk of progressing into those more severe forms of roughly 5 to 6-fold, depending on where that starts from. So what are the things that we think are contributing to the persistence of hand in patients? And so obviously there's a lot of work looking at HIV-associated factors, the virus itself, living in the reservoir, or just being a reservoir itself, that alone contribute to dysfunction in an organ like the brain? Certainly comorbidities, it's a very complex patient population filled with substance use disorder, cardiovascular disease, mental health disruption, and so among a whole bunch of other things. And so comorbidities could also be contributing to the cognitive impairment that we're seeing. There's growing bodies of evidence that stigma in particular as a social determinant of health, as well as healthcare access, the education level of a person living with HIV, and or many of the other sort of aspects of just where you grow up, the things that can be identified or associated with the zip code you live in, appear to also be associating with cognitive impairment and mental health. We and others have done some work to look at the treatments that people with HIV are taking, including the antiretroviral therapy that is certainly the life-saving medicine that keeps them alive and has turned HIV into a chronic disorder instead of what was a death sentence. But there are also a number of other what we call polypharmacy conditions in people with HIV, because they often will be taking depressants, statins, or other compounds to address other health conditions. And then there are these intrinsic factors like the host genetic factors. And so this is one of the ones that I'm going to focus on today. We have a number of genes that have been associated in varying, depending on the cohort with HIV. And today I want to explore whether that we might be able to identify another one. So the working model of HIV, as the way I think about it, is that HIV gets into the CNS. I believe there's evidence that this may be happening through T cells, through direct transcytosis, as well as through monocytes. And so I think there's lots of ways that people have proposed that HIV is getting into the CNS. The evidence is that it gets there and it gets there early. And once there, we know that it can infect microglia, and that certainly astrocytes have HIV DNA integrated, whether that is through infection and or the phagocytic activity of those astrocytes. Either way, those two cell populations can be seen being activated. And when they're in their activated state, they release a number of factors that will ultimately impact the neurons, leading to some observed synaptic damage and or neuronal death in the more severe cases back in the days of encephalitis. But we think now there's more subtle neuronal dysfunction. We also have evidence that they may be affecting the other cell type in the CNS that functionally supports the neuron, the oligodendrocyte. And by changing how myelin is being regulated along those axons, as well as persistence of having sufficient white matter, we think that that may also be contributing to some of the observed effects in patients. We also know that the antiretroviral therapy keeps the virus suppressed, which is what we need to do in terms of supporting our patients. But we and a number of others have found that antiretrovirals affect the blood-brain barrier. They affect the microglia and astrocytes, as well as oligodendrocytes and neurons. So the question becomes, what is the impact of that within the CNS of a person who's taking these medicines every day for the rest of their life? The things that I'm going to tell you about today is that among those things that either are associated with the virus or with the antiretroviral drugs themselves, activate a framework or a cellular framework known as the integrated stress response. So as I mentioned, it's a framework of the way a cell senses stress and how it handles that stress. And so you can see a number of different sort of ideas or stressors that are listed, such as not having sufficient, so starvation or amino acid deprivation, heme deprivation, infection with a virus, and ER stress, all that have sensors within the cells. And these sensors are then listed in the next line of this graph. And these sensors include GCN2, PKR, HRI, and Perk. I'm going to be focusing today on Perk. But all of these kinases, including Perk, will phosphorylate the eukaryotic initiation factor 2 alpha, or EIF2 alpha. And in its phosphorylated isoform, it downregulates cap-dependent mRNA translation. So the idea is that cell is under stress. We need to downregulate the pressure on the cell. And one of those biggest pressures on the cell is synthesizing new protein. And so you shut down the sort of massive new protein synthesis. But however, due to a sort of clever structure in the sort of mRNAs of a subset of genes, they have a number of small open reading frames upstream of their start. Those specific mRNAs will be preferentially translated at a higher rate. And this includes proteins like ATF4. And once things like ATF4 are produced, they will activate transcription of a number of other genes that have these micro open reading frames that will help reestablish homeostasis, as well as regulate the system to go back to a homeostatic state. And so today, the most important thing is Perk is one of the stresses that's the stress sensors that lives in the endoplasmic reticulum. It is very acutely linked to protein stress, and that when that is activated, we phosphorylate EIF2 alpha, and then we activate a subset of transcription factors. And those transcriptional targets that are activated specifically by ATF4 include a subset of classes of genes that we know are activated in HIV. So this includes things like cytokines and chemokines, so an inflammatory state like IL-6 and TNF alpha. Sometimes we see activation of cell death pathways like caspase-4 and CHOP. One of the main things that occurs often is upregulation of chaperone proteins. BIP is a regulator of this pathway, but so is heat shock protein 70. So these are the proteins that will come in and reestablish protein folding and protein homeostasis. What's interesting is that a number of autophagy genes are also upregulated, like ATG5 and ATG7. Perk downstream signaling also includes activation of the antioxidant response, including glutathione S transferase, NADPH quinone oxidase, as well as heme oxygenase 1. And then as well as some iron regulatory genes, so the ferritin signaling pathway. And what's interesting is Perk has a second kinase target, which is NRF2, that also co-activates a subset of these. So you can imagine during cellular stress, you have activation of a number of these sort of subcategories of pathways that we think formulate a signature, or certainly a signature that we see coming up in people with HIV. So just a little bit more about some of that work. Certainly, we have previously reported that the integrated stress response is activated in patients with hand compared to their normal counterparts. What's interesting, however, that the Perk arm, that ATF4 arm of the pathway, is really activated only in astrocytes, at least in our observations. And that will be important later. But nonetheless, we are seeing this overall activation in patients with hand. And this dovetails with reports that of transcriptomic studies that come from Donna Gabuzda's lab, where they found that also among the key signatures that formed I would say like a biomarker platform, if you were to take a sort of the transcriptomes that come up, and you take that there's 7 that when they come up, they sort of really predict that the person is likely to have hand. EIF2 signaling, or the ISR, is one of those sort of nodes that she found in that study reported by Solomon et al. And these also correspond to some work from our group looking in an induced pluripotent stem cell triculture model, where the EIF2 alpha response was induced in neurons, astrocytes, and HIV-infected microglia. So this pathway does seem to be central to a lot of the pathologic processes that are going on during HIV infection. So this led us to question whether Perk might be contributing as one of the genetic factors. And there's a number of reasons why we thought this might be the case. So the gene for Perk is called EIF2AK3. I like to call it Perk because that's easier to say, but just so that you know, when I'm saying the Perk gene for EIF2AK3 is actually the proper gene name. And the gene itself encodes a protein, and that protein sits in the endoplasmic reticulum. So it has a transmembrane domain. It has a luminal domain within the endoplasmic reticulum. The responsibility of this luminal domain is to sense misfolded proteins, to be regulated by the factors that keep the protein silenced, as well as to dimerize, because dimerization of Perk, and actually oligomerization of Perk, is essential for the kinase function. The cytosolic sign has a kinase, and this is the domain that phosphorylates EIF2 alpha. So when we looked at that, and this is a sort of picture of that protein with a kinase domain and the sort of oligomerization luminal domain. The gene itself is very large. This protein is about 1,000 amino acids. The gene is gigantic. It has 17 exons. And it's interesting because there are 3 coding genetic variants that coexist in very, very strong linkage disequilibrium. So if you inherit this SNP in exon 2, you 100% of the time inherit this SNP down here in exon 13. And there's a high percentage of the chance that you're going to inherit this SNP in exon 3. So if you look at these different SNPs, what we find roughly in the population, 50 to 70% of people have what we would consider haplotype A, or the major allele of EIF2AK3. Roughly 10 to 29% have haplotype B, which is a change in all 3 of these SNPs to the minor allele. And then a very, very small percentage of people have what we call haplotype D, which is a change in only this RS13045, the SNP in exon 3. And so what's significant about these SNPs, if you look at the fact that they are in the coding region, 2 of them occur in the luminal domain, so they can be associated with that function of oligomerization, or with the ability to bind misfolded proteins. One of them is in the kinase, very near, but not in, but very near the kinase domain. 2 of them are changes from serine to non-phosphorylatable amino acids, and so they could be changing regulation. And of course, one of them is being changed to a glutamine, but this is the one, it's interestingly, that can occur on its own. So one of the things about these changes, right, these are occurring in, the minor alleles are in basically 1 in 3 people. And so they're very common. And those 1 in 3 people live, normal lives without very clear or obvious disease. And so it should be, I just want to make it clear that these genetic variants encode functional protein that we think are fine for an individual to live with, right? So 1 in 3 of us have them on this call, and we're fine. But what we would hypothesize is that because this is a protein that functions very specifically in stress, that you wouldn't see the effects of these changes be revealed unless there was an excessive or more chronic stress. And I'll come back to that in a minute. So one thing to note, and the reason I listed ranges for the frequencies of these genotypes is that they vary by ethnicity. And so you can see that in people of African descent, it's roughly 5 to 10% of individuals have that haplotype B, whereas in your people of European descent are roughly about the 30% range, while people in East Asia have almost a 50-50 chance between. So again, with the high percentage of people having this minor, what we consider a minor allele, we think that this is, again, a normally functioning gene that may add or contribute when there is dysfunction. And this will come back in my modeling later. So why do we think this might be important, or why even study a gene that most of us have and don't have any disease from, or many of us have? The idea is that we have already reported that in very well-powered GWAS studies, this minor allele of Perk has been associated with progressive supranuclear palsy, a very rare disorder. But it is also associated with prediabetes and, interestingly, with Alzheimer's disease in a very select subset of individuals who have ApoE4 of Han descendancy in China. And so this led us to think that there might be a way to look at this as a genetic variant and understand how it might be contributing to progressive supranuclear palsy, but more importantly for us, thinking about how it might be contributing to HIV-associated neurocognitive disorder. So the very first thing we did was to look in Charter, so in collaboration with Scott Latender and Ron Ellis, we did an analysis, and this work is right now out on medRxiv. It is being reviewed. Hopefully soon we will get it out there as a full publication. But we looked at whether Perk haplotype B, those minor alleles associated with global deficit score or the domains that are tested as part of the Charter study. And what we found is that indeed all 3 of the SNPs associate with worse global deficit score. And interestingly, they all also associate with motor domain scored dysfunction. The one SNP, this one that encodes arginine to glutamine, is more associated with executive and learning dysfunction, while the other 2 are more associated with verbal domain dysfunction. And so this suggested to us that that haplotype D may be playing a role in this cohort. It is underpowered for us to be able to make any comments about haplotype D, but certainly we can say that haplotype B is associating with worse scores in this study, and certainly associated with motor domain deficits. So we then also did some multivariate analysis to look at whether, if we control for other variables, does this still associate it with neurocognitive impairment? And indeed, this variable, the SNPs do. I'm showing you only the data for RS13045, but it's true for all 3 because they highly covary in the population. So I'm going to come back to this concept. So now we have this genetic variant. It's associating in our studies. It has what we would call a moderate effect size. So what does that mean in the context of genetic variants? And so this is a graph to sort of talk about allele frequency, how frequent you see a genetic variant within the population, starting from very rare to common, the highest amount being 50%. And then we are going to then compare that with the odds ratio of developing disease. So what is the odds that someone who has that variant is going to get the disease? So on the x-axis, we have the percentage of people who have the minor allele. And on the y-axis, we have the percentage of people who have the minor allele versus the major allele, and how many of them are showing that trait. So if you have a strong association, a very high percentage of the people with that minor allele will show the trait, whereas if you have a lower effect size, a smaller percentage of them will have the trait in comparison. But the hope would be that we would identify people who would have an effect. So a weak effect size, you can see here, I'm showing the weakest effect size that it's not different at all between those with the one allele versus the other. So what I'm showing you here is the comparison of Perk B with its moderate effect size and odds ratio of 1.8 to 2, which is a significant association. However, it is a moderate effect size. Whereas you have ApoE4, which again has it's a little more rare, but it is common enough, and it has a larger effect size. BRCA1, rarer, but also a strong effect size. And then I'm comparing it to another minor allele of Perk, which basically is associated with a syndrome called Walcott-Ralston syndrome. And it is a disorder that has effects on the brain, liver, and pancreas. And you can see that this one is very rare, but it has a very, very high percentage of individuals who will present with the disorder. So this is the kind of state where we would say this is a genetic disorder caused by the gene. So you can see that where Perk is falling on this. So we are looking for moderate effect size genes that will contribute to the disorder. So we hypothesized that the genetic variation in Perk will associate with risk for hand due to subtle but increased activity of the Perk protein in cells of the CNS. So how might Perk, these genetic changes be contributing to Perk function? So there's a number of things that I mentioned. Perk to function must form an oligomer, so it must dimerize and then form multimers in order to be an active kinase. It needs to bind to unfolded proteins. It needs to have kinase activity. It needs to be stable. Perk is very stable. It lasts for 24 hours. So you can imagine anything that might change that stability could change the activity, as well as the regulation of Perk. And I mentioned when I discussed the different changes, how they might be contributing to each of these domains. But today I'm going to focus on the kinase activity. But I think exploring each of these will be important in the future. So I already mentioned that the minor alleles are associated with a number of CNS disorders, including hand, but it should also be noted that it is associated with prediabetes, lung cancer, and growth hormone deficiency. And if we looked in vitro at the kinase activity for Perk, there was a report out there from 2012 that said Perk B is more active than Perk A. They looked, but only when there was stress. So if you look at baseline, A and B are the same. But if you induce ER stress in lymphoblastoid cells, B is more active. On the other hand, we have a paper that came out about 6 years later saying, actually, in MEFs that are where we induce ER stress, Perk B is less active than A. It's actually the AB that's more active, but in reality, B is less active. So this was a controversy in the field that we sought to look at. So the very first thing we did was we purified Perk A, we purified Perk B. We took that very pure protein, and then we did an in vitro system where we added in the substrate, EIF2 alpha, and then we added in ATP, magnesium, some buffers, and we looked at how well Perk was able to phosphorylate EIF2 alpha. And so we have this antibody to phosphorylate EIF2 alpha up here. You can see that A, and in all my slides, A will be blue and B will be orange, or goldenrod, I think might be this color. And so you can hopefully appreciate that as time goes on, we are definitely seeing an increase in the amount of kinase activity in the B allele. And then, while there is a decrease in the amount of total EIF2 alpha, it's decreasing at roughly the same rate, and Perk is stable. So if we normalize the amount of phospholate to alpha to total EIF2 alpha to Perk, what we see is a significant increase in Perk activity. But again, you can see it's subtle. It's not like there's a 50-fold increase in the amount of Perk activity. It's just a little bit more active. So we know that Perk B can be more active at a very minimalistic state in vitro. But what we need to know is what happens in vivo. And so to get at this question, we made a mouse that has sequentially, we changed each of those SNPs in the coding sequence, the Perk protein that's encoded by mouse is like 98% identical to the Perk protein in humans. So these were highly conserved, and we were able to go in and say, let's change each of these amino acids to those that would reflect the minor allele. And so what we have now is basically a Perk haplotype B mouse, which we can then look at the mouse and we can look at the cells from the mouse. And so in the next minute, I'm going to tell you about 2.5 years of work by a graduate student in the lab, Shivesh Gura. And so Shivesh took these Perk haplotype mice that arrived on our doors, I'm not kidding, a week before we shut down for COVID. And over the next 2.5 years, bred them to the point where we were able to look at a couple things. So first, was there any difference between A and B in terms of pathology? And whether we looked at animals that were 2 months old or we looked at animals that were 13 months old, there was not any true overt pathology between A or B, and there was no difference by sex either. So in males and females, it was roughly the same pathologic outcomes. If we looked across those tissues at the baseline level for Perk kinase activity, Perk levels were the same. So Perk A and B had, the same amount of Perk, the same amount of EIF2 alpha phosphorylation in brain, in liver, in brain. And those were, brain, liver, and pancreas, which were the tissues we focused on. However, if we gave an ER stressor, in this case, I believe we gave tunicamycin, we over a period of time and then looked at those tissues, we were able to see that there was indeed elevated ER stress in those tissues over the baseline, but in both A and B. However, in the liver and the pancreas, the amount of Perk activation was greater. So there was more kinase activity in the Perk B liver and pancreas when there was ER stress than in the Perk A mice. What was interesting, though, that despite this change, and that the fact that the basal levels were the same, we did have 2 tissues in the brain, in the hippocampus and the cortex, where the Perk B targets were elevated, particularly BIP and CHOP. And so that led us to believe some interesting things for the future. Limitations of this model. The exonic sites are conserved, the intronic sites were not conserved. And so we were not able to introduce the intronic SNPs into this mouse. And without question, the intronic SNPs may be having their own impact on Perk regulation. Certainly thinking about Perk, mRNA metabolism is something that another postdoc in my lab is looking at, and she will be presenting on that in February. The other thing is that we focused on studying A and B and did not look at A, B together, where we might see some of that more hypomorphic phenotype that was observed in that 2018 paper looking at progressive supranuclear palsy patient fibroblasts. So for our modeling, what we're doing is we're going to activate Perk. We use 3 different things to activate Perk. These are 2 of them are classic endoplasmic reticulum stressors, thapsigargan, which it activates the circle pump to release all the calcium into the cytosol, activate ER stress. Tunicamycin inhibits protein glycosylation, and therefore protein glycosylation doesn't occur, so proteins gum up in the Golgi, back up into the ER, and end up leading to ER stress. And then we do have a compound that very specifically activates Perk. And when we can, we use that one because that really gets at our question of whether this is a Perk-mediated event. And once Perk is activated, we end up having EIF2 alpha phosphorylation, which is often one of our readouts that you'll be looking at. To look at this, we started in primary rat neuroglial cultures. So these are mostly neurons. They have a small percentage of glia. We don't get rid of the glia using aramycin C, but at least so there are some aspects there, but they are not the same ratios that you would see in the CNS. However, when we looked at in these neuroglial cultures, what we found is that if you look at time after being induced with ER stress, or in this case, thapsigargan, if you look at the Perk A animals, you have you have sort of kinase activity that follows the kinetics that we're pretty familiar with Perk, which is that by 2 hours, you have sort of a maximal activation of Perk. And then that signal decays over the next till about 8 hours, where it comes back down to roughly baseline. If you look at Perk B, you can see that there's, Perk B is in orange, and there is this elevation at specifically the 4 and 6 hour time point, sort of suggesting that the kinase activity goes up to roughly the same rate as A, but it stays on a little longer. So a little different than what we saw in our pure in vitro assay, but interest suggesting that there might be this just sort of prolonged state in which there is ER stress activation in a Perk B neuroglial culture. So that was exciting, and it was amazing that my student was able to do this very like fine-tuned long time course. We then went and said, well, let's look at the neurons alone. And when we looked at pure neurons, we didn't see a difference. So this suggested to us that there might be cell type specificity, kind of very similar to that those two publications saying that in lymphoblastoid cells we got one thing and in fibroblasts we got another. We were seeing that the neurons were not exhibiting that same difference that we were seeing in the neuroglial cultures. So Shivesh wisely went in and looked at neurons, astrocytes, and macrophages. And what we found is that indeed in the astrocytes, the haplotype B astrocytes had a greater capacity to induce B compared to A in response to thapsigargan, and so in both the macrophages and the astrocytes. And so this suggests to us that perhaps it's the astrocytes that are mediating this change in the neuronal survival that we might be looking for. So how might this be happening? So, I mentioned that there's this sort of subtle increase in Perk, and it's one of the other things we know about Perk is that not only does it responsible for dealing with stress, it is also responsible for preconditioning to stress. So if you're in a chronic stress state, you will be experiencing stress again and again. And so preconditioning is important because it allows you to mount a higher stress response and a faster stress response in a condition where you're already experiencing stress. So this is a process called hormesis. And what this shows here is that if you have a toxin or stressor, that as you increase that stressor, there is a point at which after you've already had an initial stress, you will actually induce a marked increase in survival during that time. Although, of course, there's a point of diminishing returns where there's just too much toxicity. So to put this into a real paradigm, if you pre-treat the cells with a stressor for 1 hour, you let them recover for 5, and then you induce a second stressor. And in this case, they were using HT22 hippocampal cells that were then induced with the stress with thapsigargan, and then recovered for 5 hours, and then treated with glutamate at either 5 millimolar or 10 millimolar. And what they found was that indeed at those low doses of thapsigargan, you saw a very high rate of cell death. But if you added the pre-treatment at a certain stressor of like 0.4 to 0.6, you started to see this protective response where these neurons now exhibited benefit. And actually 80% of those cells would survive in response to that glutamate stressor compared to their counterpart where they had no thapsigargan or no preconditioning. So this response was mediated in this paper, found to be mediated by Perk. And so what we wanted to do is to find out, could Perk be associated with altered stress tolerance, and that be contributing to the paradigm that we've been thinking about. So we also started with a 1 hour pre-treatment. In this case, we're using the Perk activator and tunicamycin. We then allow 5 hours of recovery, and then we add the second stressor of NMDA. Hold on. And then so here we're showing the Perk A neuroglial cultures, and hopefully you could appreciate there is a roughly 30% survival rate with NMDA in our control cells. However, if we use a low dose of the Perk activator, we see that hormetic response, a significant increase in survival over our DMSO control. And then at medium and high doses of that Perk activator, we are losing that protective effect. And this is similar to what we see with the tunicamycin, another potential ER stressor. When we looked at Perk B, we did not see this hormetic response. So it looked at least at this concentration of Perk activator, Perk B was not inducing that sort of preconditioning that would allow the cells to sort of stave off or survive in a more chronic stress paradigm. So again, this is neuroglial cultures. So our question was, is this the astrocytes that are contributing to that? And this is a very busy slide, so I'm going to walk you through it. What Shivesh did was take neurons from those Perk A animals at E16, E18, treat them with RSC, so they would be pure Perk A neurons. He then had sort of two plates going in parallel, in which at day 8, he seeded on either Perk A astrocytes or Perk B astrocytes. In this case, there's a neuron to astrocyte ratio of 2.5 to 1. And then allowed those cells to mature to day 19, and then perform that same paradigm of 1 hour of Perk activator, 5 hours of recovery, and then induction with NMDA to induce excitotoxicity. And then we looked at neuron viability. So in this case, we have sort of the neurons and neuroglia. Just to remind you, there was a minor amount of astrocytes here. In this mixed culture experiment where we're putting in the neurons and the astrocytes, there's roughly 50% neurons to 50% astrocytes. And at baseline, they look pretty similar and pretty healthy. So the first thing I'll show you is the treatment of Perk A neurons with Perk A astrocytes. And just like we saw with our neuroglial cultures, the Perk A DMSO exhibited cell death with the NMDA-induced toxicity without any preconditioning. But if we precondition with that Perk activator, we again see a significant protection. However, when we add in the increasing doses, that goes away. We see the same thing with tunicamycin, that both Perk activator and tunicamycin are preconditioning those cells to give greater survival after a stress. However, when we did Perk B astrocytes, even though the neurons are still Perk A, we again lost the ability to protect those neurons from survival, suggesting that, again, the Perk B astrocytes are not able to precondition and promote survival of the neurons. So hopefully you'll walk away with the message that we do believe that this Perk risk alleles are associating with worse global deficit score, cognitive impairment in people with HIV, as well as depression, which is a paper that was published by Ron Ellis in collaboration with us, as well as motor dysfunction and/or executive and learning dysfunction and verbal dysfunction. And then when we look at the Perk haplotype, at baseline, it's normal, which is what we expected. We don't see big differences between A and B. However, once we induce ER stress, we do see that Perk haplotype B exhibits enhanced kinase activity in a cell-free assay, in the liver and pancreas, but most importantly in astrocytes and macrophages. And when it is in those astrocytes, haplotype B astrocytes have this reduced stress tolerance, and therefore they're less able to mitigate neurotoxicity. So what does this mean? I think we still have a lot of work to do in understanding how else the Perk variants are being regulated or how they're changing Perk function. So looking at those other methods or steps in Perk activation that might be regulated by Perk, as well as looking at the non-coding SNPs that needs to be thought about. We do think the cell specificity is interesting, and then getting more deeply at how Perk is regulated in specific cell types, how that stress tolerance is mediating effects in glia versus neurons versus other cell types in the CNS. We're wondering if the Perk genetic variants might begin to form that framework where you might have a set of genes that will predispose you to developing hand that might be used to personalize treatment. And then could we therapeutically leverage that hormetic window to treat patients and protect them from HIV-associated insults? And I did try to have the names of people on the slides, but none of this would be possible without the long-term collaboration of Chala Akai Espinoza, who has been working on this with me since we started this project. I want to call out Sarah Bond, a postdoc who developed the strategy for former graduate student postdoc who's now postdoc somewhere else, who worked out the strategy for purifying Perk. Much of the work on the stress tolerance was Shivesh Gura, but that was definitely, I think, an interaction between him and Sarah that came up with a lot of those ideas, but couldn't be more proud of the work that those students did. Of course, Xinglong Shi and Noah Baraton, a talented undergrad who's now in graduate school, who contributed to other parts of the project that I didn't quite have time to talk about today, or I talked about tangentially. This was made possible by a collaboration with Jerry Schellenberg, who identified the EIF2AK3 risk variant in progressive supranuclear palsy. And then the collaboration with the charter folks, Scott, Ron, Igor, Bob and their team, as well as Asha Kalyanpour and Will Bush at Case Western. Of course, thanking the funding sources and the tremendous resources that NIH funds that make studies like this possible. Couldn't be possible without the many centers that are supported by NIH and NIMH in particular. So thank you all very much, and I'm happy to stop sharing and take questions.
Raha, do you want to field those?
Sure. So I think during the talk, Scott sent a question asking about immunometabolism, dysmetabolism, and cellular exhaustion. Do you want to elaborate? It sounds like it's a discussion you guys have had before that would now be in this context.
Yeah, well, so Scott and I talk a lot about this because we've been working on this project together also for a very long time, at least 7 years, maybe 8 years. So a long time. So the idea that Perk would be contributing to immunometabolism is absolutely, I think, spot on, Scott, for something to think about next, because we do think that it is specifically the immune cells, lymphoblastoid cells, macrophages, if you consider the astrocytes as a sort of part of the immune, or at least the inflammatory, the innate response in the CNS. We think that's where these changes are occurring. And because Perk not only regulates, sort of the response to stress, it does also appear to regulate things like lipid metabolism, as well as the inflammatory response. I could imagine this sitting right at that sponsor of the shift from adaptive to innate immunity, which seems to be one of the things that we see in inflammaging. And so I would definitely be interested in thinking more about it in the context of dysmetabolism. I think it may not be alone, because there's other ISR inducers that would be potentially PKR, because it is associated with the interferon response, is one we've thought about a lot. But GCN2 is another that would be interesting, simply because, and HRI, because they think they are associated either with iron metabolism or with amino acid and glucose deprivation. So thinking about the ISR as a broader framework, I think, is also valuable. So I think Perk for sure. I think the other kinases would be worth considering too.
The glycolysis and glucose part of it too aligns with some work that others have done, but particularly I think the Hopkins group with insulin resistance in the brain and administering insulin transnasally and so on. So it suggests that a certain combination of factors that developed independently that may align and lead to some therapeutic advance. So it's nice to think about, but a lot more work needs to be done. But
I agree, lots more work. Well, and what's interesting, and I didn't have time to talk about, is we do think this models pre-diabetes. So if we look in the pancreas, what we're actually seeing if we induce ER stress is that those cells are way more vulnerable and they die, while the liver is actually just activating a stress response to protect itself. The pancreas are dying. So they are far more susceptible. And that aligns with, other knockouts of things that we know are like knockout of Perk. There's a pre-diabetic phenotype when it's knocked out. So it's interesting that the Perk B might be giving some phenotype like that. So yeah, there's a lot of connections to think about. Thanks, Scott. That's a great thought.
Question?
Yeah, so then it looks like Jules asked about stressors and whether or not they include ART neurotoxicity, low CD4, detectable viral load. And it was answered later that it was accounted for in multivariate analysis. But Jules, I don't know if you had a separate question following up from that.
Yeah, I'd like to know if those were all stressors that you referred to in the stresses you identify as what's needed to cause, actual clinical cognitive impairment, if those stressors are all relevant to that. And I'm wondering, in addition to that, social determinants of health, lifetime discrimination, traumas like that. And then the second question is, so if these all stressors could be the reason that differentiates those people with HIV that get hand from people that don't get hand, why aren't we really seeing higher rates of hand? Because everybody with HIV really has these stressors. And as they're aging, the hand rates should be going up. And they seem to be somewhat stable. And then thirdly, this could be, these stressors could be increasing the risk for Alzheimer's in people with HIV. So there's a lot of questions. Thank you.
Lots of great questions. And so I'm going to try to remember them all, but I think certainly the social determinants of health and whether, I think the issue we have in terms of like separating out and doing the analysis, as Raha mentioned, we did our best to control for the viral load for the CD4. I don't know how well social determinants of health, I think we're still getting really a grasp on how to incorporate that into our modeling. Certainly stigma, I just saw a really great talk at ISNV on the importance of stigma in people with HIV in terms of contributing to mental health and other disorders. And so I think that the answer to that one is still we need a lot of work, and I'll say we need bigger cohorts and better well-defined cohorts. And I'll say one of the other parts of that in terms of the genetic risk, why some people with certain just social determinants of health will be at greater risk than others. It's the same that we have with HIV. And so you really need to think about all those possible contributors. And I just don't know that I'll say mathematically, I don't know how to do that yet, but I think it's something that, we're trying to get at. I am definitely looking at this concept of if we knew that there was a genetic variant that we would be able to target. And because we do know that this one also is downstream of some of the ART toxicity that we've studied as well. Could we do something that would prevent or help a subset of individuals who have that? But it is very possible that this Perk variant may also be associated with other things independent of HIV. So we're seeing depression coming out in our thing. What I don't know is, does it come out in a larger cohort of depression? And we just haven't been able to focus on those other outcomes. The Alzheimer's disease question, I think, is a great one. Certainly, this variant is already associated with Alzheimer's disease. So the question is, will people with HIV who have this variant be at higher risk for developing Alzheimer's-like changes? And so I think that's something that we need to look into. You had another question in the middle of that sandwich that I don't remember, Jules. So I'm sorry.
Yeah, so all these stressors are all very common amongst almost everybody with HIV or lots of people. So why aren't we seeing increasing rates of hand as the population is aging now into their 50s and 60s?
So I think there's a couple things, but I'll leave this to my clinical colleagues who probably know this far better than I do. But my understanding is there are a few things that have changed in terms of clinical care. So I think the first is we're getting people in clinical care earlier, and we are doing a better job of maintaining those who we get in care on antiretrovirals and having the positive treatment. And so I think that is, without question, the right direction for things to go. On the other hand, we do still have a number of individuals infected before that have different risk factors that I think have persisted with hand. And so I think the data for me is still out there. I'll admit that, I'll just share the historical evidence that in 1996, we thought we found combination antiretroviral therapy and hand and HAD would go, and HAD went way down, right? But, and what we saw was this increase in prevalence as people were able to survive longer with the disorder. So you have a percentage of individuals who may already have pre-existing conditions versus those who are developing new conditions. And so, but it took us, I think, 10 years before the data started coming out to show that despite this, cognitive impairment was persisting. But I can't tell you why, I think we still have a lot of work to do to understand why some people, and whether they have with HIV or those that even without HIV are more susceptible to some conditions versus others. I think this is still a really challenging question for the field to understand, especially in the context of mental health. I'll point to things like biotyping, the fact that many of these things are, they're a diagnosis based on a phenotype, and they're therefore heterogeneous in terms of their actual cause and outcome. I saw Scott poke his face on. Do you want to add to that, Scott?
This is about resilience.
Well, you did great, but it was a little bit about resilience, but just the very data that you presented, right? I mean, that not everyone has those genetic risk factors. So if you have, for instance, those genetic risk factors plus, the terrible social determinants of health, you're more vulnerable to injury from that, from chronic injury from that. So if you don't, you're not. So, the data you presented, Kelly, on genetic determinants, in part answers what Jules is asking. So,
Yeah, it's not the whole answer, but I agree. I'm hoping that ends up being part of the answer. So that's one of the things why we want to work on this, Jules. So thank you very much for those questions.
Well, he also commented that the newest version of Charter is doing a better job of collecting more data on social determinants of health. So maybe one way we can rephrase the question isn't what contributes to cognitive dysfunction, but maybe what's protecting you from declining. So
that's a great reframe.
I've gotten a lot of questions here. People are very engaged.
Good,
So,
I'm excited. I gotta download these questions so that I have them.
And I can copy them and send you a copy, and maybe Cassidy can as well. But I know that you wanted feedback, and I hope that these next questions are helping with that. So Dr. Kashanji specifically asks about the specificity of Perk A and B, whether that has changed, and whether you've thought about doing phospho-LC-MS and looking for broad substrates in cells.
So we're right now working on the IP to do the LC-MS. Perk is very big, and so therefore IPing it is more challenging than some other things but we are working on that. So as like literally as we speak. So the specificity, can you explain specificity in terms of whether it's targeting EIF2 alpha, like thinking about the substrate specificity? Is that what you mean? Yeah, that's a great addition of idea.
So thank you, Kelly. That was great. It's many times I see this where when a kinase is either overexpressed or it has acquired new partners where it changes its location from a monomer dimer to a tetramer or sitting on a scaffold such as RNA, all of a sudden the specificity changes. So now it requires less ATP, and now it will start phosphorylating a bunch of substrates on the membrane that it's not supposed to, or even on the ER. So it's just that its behavior changes and kinetics of phosphorylation changes all of a sudden, which makes it a perfect therapeutic target, right? So it may be that maybe the size changes or even the profile of phosphorylation changes, and that could give you a broader spectrum of coming up with therapeutics.
No, it's a great idea. I love that idea, Fada. It's wonderful. Thank you.
So I see Dr. Bush is showing her face. So maybe you want to ask your question yourself, or I can read it out, whatever you like.
Terrific talk. Really enjoyed it, Kelly, as always. So the question is, did you look at microglia and their phagocytic activity? And since ER is so closely attached to the mitochondria, the MAMs, have you looked at and comment on something on those? And plus, looking at ferroptosis or else inflammasomes, how does this all relate to? I mean, it's pretty complex.
Yeah, no, it's really complex. And so phagocytic activity we looked at in macrophages. We did not see it affect phagocytic activity, but I will tell you the phagocytic activity of the macrophages was getting the kinetics down is challenging, but we did not see it affect phagocytic activity specifically in those macrophages. So I think the inflammasome is something that we are looking at whether how much is changing the inflammasome. We have seen it change IL-6. So what does appear to change is the amount of IL-6 that's secreted from the astrocytes in the macrophages. We haven't looked at microglia yet. It's, because we were able to get the macrophages faster. But microglia definitely we need to dig in, but I would expect them to be similar to what we were seeing in the macrophages. You had another question, Shilpa.
Have you looked at the mitochondrial membranes?
Oh, the MAMs. So actually I just recruited a student to my lab, and that's her project that she wants to work on, which is looking at the MAMs and definitely because there is a mitochondrial associated ER stress. And so understanding the difference between mitochondrial ER stress versus ER stress, right? So, and I think it is going to be related to these MAMs. So well, she's working out some technologies right now, but I think it's also, again, a really great question and direction.
Thank you. Great work.
Thank you.
Thank you for asking about the mitochondria. That was going to be my question too.
Well, I mean, so if you've heard other talks from people in my lab, or certainly Jonathan Geiger and I are thinking a whole lot about the organelles as a whole. And so while I spent a lot of time talking about the ER, I think about them all sort of acting together: the mitochondria, the ER, the lysosome, the Golgi too. Although I'll admit I have less tools to understand and look at the Golgi, but I think we need to be thinking about them all. Like when one's affected, the others are all affected, and that like feeds back to autophagy or forward to autophagy, right? I mean, so this is something that, you know, I presented in a way that's I can get at a logical outcome in one sort of linear story, which is how I think. But I also am aware and constantly trying to put all of these things into a broader context. Unfortunately, that makes me unfocused, but I think it is fun.
Well, I think it's just complicated. So, I mean, like the ER and mitochondria are both calcium reserves, and the calcium through PAP-Scargan was one way that you were activating Perk. So I think I really liked how you laid it out, because it helps to kind of split it up and see where you can target and what you can find there and how to relate it back to clinic.
Cool. Thanks. I appreciate that.
I like flowcharts. What other, there was another question. Oh, so Rob Paul, I don't know. That might have been in response to the protective factors comment I made, the complex interaction, everything's just so complicated. And then there was one last question from Jules about an age threshold, I guess, in the clinical part.
Yeah, we're not looking at, we didn't discriminate by age, so we just used it and accounted for it in the multivariate analysis, as I think Scott has alluded to. So we didn't cut it off by age, we just simply looked at everybody that was in the cohort. And so we haven't looked at whether there is an age-specific effect. And I think the cohort might not be big enough for us to do that, but maybe that's something for me to discuss with Scott and Ron afterward. But I think, that was not something that we looked at, but I think it's an important point. And I think it is why these cohorts are so critical to continue to follow, because like one of the things that, they did a GWAS with the charter, but it didn't come up with any of the hits, but I would never have expected it to. The GWAS that gave us the hits in Alzheimer's disease had 22,000 genetic signatures. The GWAS from charter has a little bit over 1,000. So, I mean, we're just not powered sufficiently. And I think that's really important with the new NIMH guidelines that are out for, looking at genetic factors. So there's new guidelines on this that I think we need to really think about as a field, because it's going to be important that if we are going to leverage our understanding of genetics or the transcriptomics that are out there. I mean, it's the work from Donna Gabuz that really comes up with, like, I would say, a signature of 7 transcripts that for her will accurately predict in a machine learning way who will have hand in a population that she's studying. So if we're going to leverage that, we just need more power. So that was my soapbox, my ending soapbox statement.
That was all very wonderful. That was a great talk, and I think great interactions at the end and answering questions. That was the last question. Does anyone have any more? I know we're out of time, but maybe I saw Dr. Gendelman show his face, so I thought maybe there might be a question, but maybe not. Oh yes, happy holidays. As a reminder,
I will ask, since you mentioned my name, I have to at least ask one question. First of all, Kelly, that was great, as always. I guess the question that is kind of I'm just struggling with, and maybe you can help me with, obviously with those lapses in my words, is where are we going to begin to unite these pathways beyond hand, and what are we thinking about the common changes, whether it's the inflammasome or the microglial response, the interactions between the astrocyte and the microglia, but there's so much commonality amongst many of these diseases. and it includes the issues of the blood-brain barrier and how proteins are drained, how signaling pathways go from the CNS to the periphery and then periphery back, and obviously trafficking. Maybe just in the, because we don't have a lot of time, but maybe you can help us in not only where we go next, but how we go next.
Well, I think you've just tasked me with something that is a much bigger job than I can do in a minute at the end of a seminar. But I think we do need to have more. So my personal feeling is the way to do this is through collaboration. And that's obviously been the sort of approach I've used. That list of collaborators is just for this project. I have collaborators for other projects. Like, to me, the way to move it is to have people who really have the expertise in each of those things and bring them together. Because what I find is, in graduate education, as a person who does that for the other half of my life, is we teach them people to think deeply, but there's a huge burden knowledge on this generation. That's our burden knowledge now, right? And so it's how does one person know everything? It's pretty hard. And so it's better if you can have multiple people thinking about it. And that does mean to me that we need not just these sort of community, when I put in a grant, there's usually at least 2 or 3 other investigators with me, so I can have all the expertise I need to answer the question. But I think that means we need more teamwork, and that teamwork would be better if it was cross institution. It's nice when you can do it all within your institution, but not all institutions have the ability to do immunometabolism, mitochondrial work, ER, I don't know, maybe you're all at that, but I find Penn is an incredibly rich place, and there's still just not everybody here I would need to do that work. So I really think that finding a way for us to have experts come across the disciplines and be able to talk about it and bring it together, but it does mean that I really believe strongly in team science for the future. So I think that's a cheating answer to your question, Howie, but because it doesn't give us any scientific content direction. But to me, it is about having the people who are the experts come together and think about it. The other part is machine learning, which I'm still myself learning. And so I think that, finding the better ways to get machines to interpret our data in ways that bring them together. Because what happens when I get transcriptomic data back is, everyone in my lab goes, "Oh, Kelly, your favorite pathway's there." That's the first thing they say to me. They completely bias my unbiased data. And then I try to look at it in an unbiased way and think about the other things, but I can only do so many things. Even with the one project, we're not doing everything we could or should be doing, right? Which just, we have to do what we're capable of doing in the moment. So I think that's what makes science fun. And so I don't want to leave that as a down thing. I think this is what makes it fun. And the stronger that we are working together as a community, I think the better we are in terms of being able to address these questions.
Thank you. Thank you very much.
Yeah, thank you. And I see that Leah emphatically agrees with your portion about team science.
100% team science. No, but Kelly, amazing talk. This was awesome. And the discussion, and I think that's what's also part of it, right? So having these forums and having these discussions, I think also helps. So this is great that, today, there was a really nice group of folks and everybody was really active. But yeah, so we look forward to picking this back up after the holidays. We hope everybody has a happy holiday. Get a little bit of rest if you can. And yeah, we will see you guys soon.
Sounds
January
great.
10th.
Thanks, everyone.
Bye.